My name is Chris Armstrong. I'm the Director of the Office of Chief Scientist and Engineer. I'm acting Chief Scientist and Engineer at the moment, but I'm not the new Chief Scientist and Engineer. They're yet to be appointed. But first of all, I'd like to welcome you to our first breakfast seminar for 2018. We're starting off the year with a wonderful presentation today from the current New South Wales Scientist of the Year, Professor Gordon Wallace from the University of Wollongong. But before I start, I'd like to acknowledge the traditional owners of the land, the Gadigal people of the Eora Nation, and pay my respects to elders past, present and future. I'd also like to acknowledge guests today, particularly our speaker, Professor Gordon Wallace, AO, parliamentarians, and thank you for allowing us to have these presentations in your lovely house, members of the Diplomatic Corps who are here today, members of industry and government and regular guests. I'd also particularly like to acknowledge Professor Mary O'Kane, who's actually in the audience up the back. She's the chair of the Planning Assessment Commission. This is our first breakfast seminar without Mary as Chief Scientist, and I'd like to take this opportunity on behalf of the Office of Chief Scientist and Engineer to publicly thank her for her many years of leadership and friendship. She's been amazing. So Gordon is a leader in the field of electromaterials, and he's pioneered the use of nanotechnology to develop organic conductors that lead to new materials that have use in energy conversion, energy storage, medical devices, and a number of other applications. The subject of today's presentation is 3D printing, bioprinters, clinical application, which will cover some of the work that Gordon has been recognised around the world for. The fabrication of bionic body parts and the development of plastics that conduct electricity. This work has dramatic implications for human performance, including the possibility of fusion of bionic limbs with nerve cells to allow them to be controlled by the brain. Gordon's recent developments include an inkjet printer that assists nerve cell generation with the potential to repair damaged spinal cords and heal hearing damage. Gordon is a Business Event Sydney ambassador, and he's also been the recipient of numerous awards and honours, including the Fellow of the Australian Academy of Sciences in 2007, ARC Laureate Fellow in 2011, Eureka Prize for Leadership in Innovation and Science in 2016, Officer of the Order of Australia last year, and of course New South Wales Scientist of the Year late last year as well. Gordon's published over 900 refereed papers that have attracted more than 35,000 citations. It's such an impact. He has supervised almost 100 PhD students and mentored over 50 research fellows. As you're about to hear, Gordon's a passionate communicator and is dedicated to explaining complex scientific advances to both scientific and broader communities. So please join me in welcoming Professor Gordon Wallace.
Thank you for that kind introduction, and thank you for being here this morning. I do appreciate the opportunity to share some of our research with you. I should start by explaining that I have a Geelong accent. When my parents immigrated to Geelong when I was 14, and I had a straightforward Belfast accent. But now, as you'll hear, it's become quite a hybrid. And I say this right up front because the poor people down the back are recording this lecture, and I neglected to tell them they might need to use subtitles as we go through. But let's see how we go. I'm sure they'll be prepared for anything. So I'm from Wollongong. It's just down the road. A spectacular place to live and a spectacular place to work. We have dedicated research facilities as part of our ARC Centre of Excellence for Electromaterial Science, which are located 180 metres from the beach when the tide's out. You can come down there and explore the whole vicinity and indeed explore our laboratories anytime that you would like to do so. We're the headquarters for the ARC Centre of Excellence for Electromaterial Science, but it does involve a number of other universities around our country and a number of other universities around the world. And as the name implies, our focus is in the discovery and development of new electromaterials. And I want to just show you why we're so excited to get into 3D printing, 3D bioprinters and clinical applications, because of some very fundamental research that we were able to undertake in the area of electromaterials science. It's been recognized around the world that even the most fundamental electromaterials, materials that conduct electricity, that transport charge from one phase to another, even the simplest of those materials, as found in a conventional battery, takes on extraordinary properties as we delve into the nano domain. As we go through the micro domain, the size of living cells, and 1,000 times less than that down into the nano domain, even what we would consider very boring materials all of a sudden become very interesting. And you know, there can't be anything more boring than a sheet of carbon, lead from a pencil, right? If there's one thing you've forgotten from your chemistry days at high school, it's carbon, right? It's one of the most boring elements that we can think of, and yet essential to many aspects of life. And who would have thought that if you get down to a single sheet of carbon, graphene, that we uncover these amazing properties, that it can be stronger than steel, yet incredibly flexible, highly electrically conducting, highly thermal conducting, and also we can tune it to be biologically active. And so there's some very fundamental chemistries here, some very fundamental and yet simple discoveries that we're able to build on as we venture into this world of 3D printing and bioprinting. And the fundamental and simple chemistries really are simple. So where do we get graphene from? Well, we get it from a mine. We take a lump of graphite from a mine, and we do some very simple chemistries, the sort of chemistries that everybody loves because it explodes things. So you explode this bit of graphite into single sheets of graphene, and at the molecular level, that single sheet of carbon has these red oxygen atoms on it. And so it turns out that tuning the properties of graphene really depend on something very simple, and that's our ability to control how much oxygen is on that sheet of carbon. Because as we take more and more of those oxygens away, without compromising the size of the sheet, that's also important, then we get better electrical conductivity. But as we take more and more away, we also compromise the processability. So it was a need to balance the chemistries in processing graphene. It was one thing to know that it has amazing properties in the nano world. It's another thing to think, how do we take those properties from the nano world back into the world that we live in? How do we extricate those amazing mechanical and electrical properties and turn it into structures that we can actually use? Well, understanding this fundamental chemistry of how you can tune the balance of that carbon to oxygen ratio, and in fact localize the balance. Imagine if we could just put the oxygen atoms around the edge of that big carbon sheet to give us processability, and yet take them away from the bulk of the sheet to give us electrical conductivity. Well, it turns out that we can. And we can use those chemistries then, not only to discover nice properties, but to turn it into real devices. In this case, turning those single sheets of carbon into a carbon fiber electrode with amazing properties in terms of mechanical and electrical properties and biocompatibility. Compatibility that enables us to insert it directly into the brain so that we can record neural activity in the brain. Now that's the most significant advance, right? Of course, the fundamental advance in terms of the chemistry of the materials was incredibly exciting to us, but being able to balance that and make sure that we could keep it processable and make a real device out of it is what really gets us incredibly excited. Now, this isn't much different from carbon. You can see, most of you will look at this and say, it's still predominantly carbon. It's still organic in nature. We put some extra elements like nitrogen or sulfur in, and this enables us to, as you heard in the introduction, generate these polymer plastic materials that conduct electricity. Polymers are generally electrical insulators. Here we have a polymer which is highly conducting, or can be highly electrically conducting. It's also organic in nature, so it's biocompatible. In fact, we can even make it more biocompatible. Over the years, we've developed techniques of synthesis where that innocuous A- that you see on the screen can really be very important. It can be a biologically active polymer. It could be an antibody. It could be an enzyme. In fact, it could even be a living cell integrated into this polymer structure at the point of synthesis. So we were interested, and others joined with us, and in fact others drove our interest in many ways, in seeing how these could interact with biological systems as well. So we were taking nerve cells from the cochlea of a guinea pig. This cochlear explant contains those living nerve cells. First of all, we had to prove that they were compatible with that new surface, and they were. So the viability of their survival was high. They could proliferate on there. And then the question was, could we communicate with them? Just like as we do, or others do, with the cochlear ear implant, where you're stimulating nerve cells in order that people can hear. Here's where this starts. This starts on the bench by generating new electromaterials and testing how effectively they interact with living nerve cells. So we were interested in building proteins and biological activity into this electrode, and then doing electrical stimulation to see if we could promote those neurites growing out from the cell body. Could we get longer neurites? Could we get more neurites growing from that cell body? And that would be an indication of our level of communication. Well, those experiments were done with Professor Graham Clark and his group. Graham, of course, maintains, even today, a very strong interest in the development of new electromaterials for the cochlear ear implant. What we found is that we could get effects just from direct electrical stimulation. We expected that. The cochlear ear implant works, so we expected that effect. We could also get an effect from the presence of the proteins, but the combination of them delivered a synergistic effect that we hoped for and sort of expected, but we're very pleased to find. So we could build now an electrode which was highly effective at communicating with nerve cells, but more than that, it was also biologically active. And it was active at the point of most concern, and that's right at the electrode cellular interface. So we've gone on to build on those studies in recent years. In fact, just last year, I think we published this work. And this again uses those amazing electromaterials. One of my colleagues, Professor Xufeng Huang, looked at that data that we generated with Professor Clark, and we had measured the length of the neurites, the number of the neurites. His interest was in the degree of branching in each of the neurites. And there was a reason for that, because he had worked for many years on schizophrenia, and he had determined that the early onset of schizophrenia really starts with a dieback in that degree of branching. And so Xufeng approached us and said, can we test this somehow related to schizophrenia? Same sort of experiment. We've got the cells on that electrode substrate, that organic conductor, and we're going to stimulate it and see if we can bring back the degree of branching in cells from schizophrenic mice. Now, I don't know how you know a mouse is schizophrenic. And just before anybody asks me at the end, that's the answer to that question. But I do have Xufeng's number on my phone here, so we can give him a call if absolutely necessary. But anyhow, these cells, the mice have got particular genes knocked out of them that are related to the development of schizophrenia. And if you do the experiment, you can see that with those cells just sitting on our new electromaterial, you do indeed see that degree of dieback in the neurites, the degree of branching decrease, and this was an amazing finding. Now we found that through electrical stimulation, we could restore that degree of branching, we could restore that neurite activity. Now the reason for that we're still investigating, but it does restore those symptoms that you'll see in the early onset of schizophrenia. The next slide's got the data, but I'm just going to flick through it because I've told you the story already. Essentially, we're restoring that degree of branching and degree of neurite outgrowth. So these sorts of findings, and working with Professor Graham Clark, of course, starts to attract the attention of the rest of the clinical community. Others who might think about how they could use these advances in electromaterials to tackle real clinical challenges. And one of those was with Professor Mark Cook, who was interested in new electromaterials that are implanted into or onto the brain in order to monitor and then counteract epileptic seizures. And with Professor Peter Chung, an orthopedic surgeon, who was interested in restoring some of the function he may have had to remove for some reasons during a surgical operation, whether that be nerve repair, muscle regeneration, or bone regeneration. So you can see that to get into what these people are interested in, including the cochlear implant with Professor Graham Clark, we had to be able to make devices. We had to be able to make those things that would go into the brain, or make those electrode structures that we could test on the bench. And sometimes the complexity of those devices is really quite high. Let's have a look at the nerve regeneration conduit with Professor Peter Chung. So we knew that these graphene fibers, or those organic conducting fibers, can positively interact with nerves. We subsequently did some work with nerve fibers and showed that we could make the nerves grow along that fiber, so you could actually control the direction of nerve cell growth, and of course that will help with reconnection of nerve cells. We also knew that we wanted to release particular proteins to facilitate that regeneration process, so we need to build those into the structure. And more recently, as you'll hear towards the end, we also want to incorporate stem cells into that structure to facilitate the regeneration process. So that's a complex structure. So we started to make those fibers individually. We would make the graphene fibers, we'd make the fibers containing the proteins, and the fibers containing the cells, and then hand assemble that into the conduit and wrap it up in this three-dimensional cocoon. So that's not even an efficient way to do research. Never mind an efficient way to get to a practical solution to this problem. So we were challenged some years ago now, working with Graham Clark and those other two gentlemen that I introduced you to think about different ways that we could assemble these very complex structures. Conventional manufacturing was not going to do it for us. These materials are not amenable to the practices we encounter in conventional manufacturing. The 3D additive fabrication does provide some promise, because here we can adapt the inks to use our materials. We can use multiple materials and we can build things up layer by layer. So materials that are not compatible with traditional manufacturing, all of a sudden you can print soft conductors. You know, you can print the materials containing the bioactive molecules, and maybe you can even print the materials containing the living cells. So 3D printing just changed the way we think about making things, and it changed the way we engaged in our research programs. Because 30 years ago, embarking on a material science research project, the goal would be to discover the material, demonstrate some effect on the bench in an isolated instance, and move on. It was somebody else's problem to take it and turn it into a structure, into a device, into a real application. And that's because the manufacturing challenge was just too big. You know, you had to convince someone that to change a whole multi-million-dollar manufacturing plant in order to make something based on your material. So 3D printing changed it all, flipped everything on its head. Now, not only could we create these structures, we can create them in the research laboratory. We can discover new materials one day, develop the ink, create the structure within a number of days, and do that alongside the stem cell biologists who can characterize and utilize that final structure. So 3D printing has had a dramatic effect. It enables us to think about protocols that will take all of these advances in materials and cell biology and integrate them into a single and practically usable device. So I mentioned that we need structural materials. So the electromaterial is critical. It provides that conduit for electrical stimulation, but alone it doesn't make it. It doesn't cut it. It has to be combined with something that will give us appropriate structural support and even enhance further the biocompatibility of those materials. So soft materials, or hydrogels as we call them, as the name implies, they contain mostly water, often more than 95% water, very much like the jelly you'll find in your fridge. You can pick it up, you can handle it, you can cut it, you can make devices out of it, but that high water content makes it highly compatible with living cells. So we've developed a whole inventory of new gel materials that can be 3D printed. And now we're getting close to that pointy end of application. We're starting to think about, well, what's really important here is not just the fact we can do it, but the source of these materials. Where do they come from? How reliable is the source? How controllable is the source? Well, one element that we're putting some investment into is relying on seaweed as a source of those materials. A lot of those biologically active biopolymers can be extracted from seaweed. They support the living cell growth. We can render them printable, and we can work along with seaweed farmers in Australia, one in particular, to make sure that we control the source of that seaweed and control the reproducibility and the quality of that source. This is Dr. Pia Winberg from Venus Shell Systems. She has a seaweed farm in Nara, not very far from where we are in Wollongong. And Pia is no ordinary seaweed farmer. She farms with quite exquisite precision. feeding the seaweed a carefully controlled diet, harvesting that seaweed at carefully controlled times, and working with us and others to make sure that the extraction procedure to get what looks like quite complex biomolecules at the bottom there ensures reproducibility and integrity of the material every time we go to develop a bioink and to use it. Even being able to print those structural materials has quite an effect. Even if we don't combine it immediately with the electromaterials or with the bioactive centers. This is some work we've been doing with Professor Peter Chung in cartilage regeneration. Now, this was a recognized therapy. It was recognized that you could take adipose stem cells from a fat part behind the knee, inject them into a defect in the knee, and the hope was that they would generate cartilage. And I understand that sometimes they did, but sometimes they didn't. And that's not really, in retrospect, surprising, because we weren't controlling the environment of where those stem cells and how those stem cells were being delivered. So a very simple advance in being able to 3D print those structural materials. This one's based on chitosan. It's a derivative from crab shells, which again, naturally occurring. And if we can match the mechanical properties or the distribution of the mechanical properties with what one might encounter in cartilage, and we can protect those stem cells during the therapy, then you would think there's a much better chance of regenerating that hyaline cartilage. And indeed, that's what we've shown, that you can take those same stem cells and instead of just directly putting them in the defect, introducing them into a scaffold that provides the mechanical properties and the protection of the cells during development is a much more effective way to generate that cartilage. Then there's other components that we might start to think about as we get more sophisticated. I've already mentioned graphene and our organic conducting polymers. So we've developed inks. And again, it was just by simply controlling that chemistry right at the start when we process the graphene from graphite, we've been able to develop inks that are either inkjet printable or extrusion printable into 3D structures. And similarly with the organic conducting polymers to develop inks that are able to be inkjet printed or 3D printed into those structures. So because we were thinking right at the start of the development of these materials compositions, and we had an eye towards the ability to process and fabricate, we were able to tune the chemistries not only to enhance the functionality and enhance the inherent biological properties of these materials, but to make sure that we maintained, and in some cases even enhanced, processability and the ability to print and 3D print. Perhaps the most challenging component, though, is to print bioactive materials. So they could be the proteins I mentioned before, or biopolymers or drugs. They're relatively simple to print. We can protect even delicate proteins. We can wrap them up in other micro capsules that consist of polymers. We can add those into the ink, and we can 3D print those with very high survival and high integrity of the molecules after printing. But as you can imagine, printing a living cell is a little bit more challenging. So let's think what we're asking this cell to do. We're going to put it in this is an inkjet experiment, so we're going to put it in an inkjet reservoir, which, by the way, initially we bought from Officeworks. An inkjet reservoir, but we did manage to get the other ink out first. That's very important if you're going to try this. The cells don't like the Officeworks ink. And of course, I shouldn't say this because it's being recorded. Yeah, we bought it at a store, I won't mention the name of the store. But anyhow, we did figure out how to clean out those inkjet cartridges and put our bioink formulations in there. And then they have to be stable there. So you want these cells to stay dispersed in that ink for a reasonable period of time and to stay alive. And then we're going to squirt them through this amazingly small reservoir at an incredibly high velocity and hit a surface at the other end, bounce off that and survive. And cells are fairly delicate little components, little entities, so we were asking a lot. But again, it was a fundamental discovery that made this possible. But what we found is that some of those biopolymers that you can get from seaweed or other natural sources are actually what we call shear thinning materials. So that is, when it's sitting there in the reservoir, it's got a consistency like honey, and so the cells don't settle. And yet when we put it through the reservoir, because it's under shear, those forces that it's encountering becomes like water. So it absorbs all of that force, and then it gets to the other end, and it comes back like honey again. But so here you have a material, a fundamental discovery of a material which has a high affinity for cells, but yet it has this combination of properties that mean you can also 3D print with it. Now, that sounds like we did that in about an hour. It took about 5 years, and a couple of the students working on it are still recovering in therapy. But they are recovering, and it's been a great contribution to science. And so we have been able to print living cells with very high viability, nerve cells, muscle cells, and you'll notice with great precision as well using inkjet printing, where you can put one cell next to another. But some of the applications require high cell density, so inkjet printing would not be appropriate. So we developed an extrusion printing technique, again using a naturally occurring polymers that we modified to make them printable. And this is now printing that cartilage for cartilage regeneration, sorry, this scaffold for cartilage regeneration, but now it contains the adipose stem cells. So the first experiment I showed you, we printed the scaffold and then introduced the stem cells. Now we're printing with the stem cells. And of course, the final clinical protocol would be printing with the patient's own stem cells into this particular structure. And again, we get very high survivability and very good development into hyaline cartilage using that structure. This did require a little modification though. Using this printing protocol actually uses light, and we used to use UV light. We've shifted it somewhere into the visible, but use light to induce that phase transformation. So to take it from a flowing gel as it came out of the tip of the printer and turn it into something which was more like jelly and would retain that structure after printing. That was okay, but we did sacrifice some of the stem cells or some cells during that printing process because of that protocol. So this is where the interplay, the interdisciplinarity of this whole area is critical, because just developing an ink, a biomaterial, or just understanding the cell interactions, that's all great. But this particular solution came from our mechanical engineers, from our mechatronic engineers. And the idea was quite simple, really. Well, why don't we isolate the cells in the core of a printed structure, and we surround them with this shell that will have all of the mechanical properties, but it won't compromise the viability of the cells in the core. So we set about designing and building a coaxial 3D printer in order to do that. And you can see that we've been successful in doing that. It's really quite a complex print head. That's really where the big advance was, in the fluidics in the print head. And you can see that print head there, where you've got pipes inside pipes, down inside a metal print head. This was created using 3D printing. So this is a 3D metal printer. So not only are we having advances in these practical things and the fundamental explorations we talked about before, the ability to realize advances in new printing technologies and new prototypes is actually enhanced by today's existing 3D printing technologies. And you can see you can build all sorts of microfluidic channels inside those 3D metal printed structures. You can't make this structure with conventional manufacturing. So not only has 3D printing said to us we can accommodate a much wider range, a bigger inventory of new materials, we can actually create structures even with existing materials, well-known materials. We can create structures that you can't create using conventional manufacturing. And I'll just diverge for a moment because I want to say a little bit about the achievements of others in this 3D printing of metals, and particularly here in Australia. Anatomics is a company based in Melbourne, and Anatomics have done some amazing things with 3D metal printed structures that are being used in the clinic today. That 3D printed heel was implanted by Professor Peter Chung, and look at the complexity of that 3D printed sternum. 3D printed in titanium with all of the flexible ribs being able to connect to existing ribs. And these are being designed and created here in Australia, where we're leading the world in this area in terms of complex 3D metal printed structures for medical implants. So back to the Biopen. I've learned in my association with clinicians that they're never satisfied, that they always want something that yet don't quite have yet. And so, we discovered this ink. We showed Peter and he showed us on the bench. We can print the structure, adipose stem cells, the cartilage. Now, whereas, okay, well, we need to print with the patient's cells. Okay, we'll do that. So we print with the patient's cells, And then the final request was, well, that's all great, but I need to be able to print into the defect. I don't want to print the scaffold on a bench and then put it into the patient's defect. I want to print with their stem cells directly into the patient's defect. And so therein the concept of the Biopen was born. Could we create a handheld 3D printer that would be able to print directly into that defect? So we used our coaxial concepts and we built that handheld 3D printer. And some work published recently, Peter and his surgical colleagues have shown that there really is a significant improvement by printing directly into the defect compared to printing on the bench and then transplanting the scaffold into that patient. So that journey continues. You know, we continue to work on all aspects, as we do with all of these particular projects, of ink and cells and hardware. I don't usually put the picture of the robot up if there's a lot of surgeons in the room, because I tend to get a lot of pushback on that. But this looks like a friendly audience, so I'll throw it up there. And so you can imagine that a robot, imagine a robot cutting loose with a Biopen. We get really excited about this. Imagine the structures you can create. You can even create structures that a normal 3D printer can't create because of the movements axes that you have on the robot. But we won't release this too much into the public till we've got it right. I'll get my surgeon colleagues on side. And then other advances. And so the Biopen, we've been fortunate recently to attract the interest and support and commitment of Mother Son Innovations, an automotive component manufacturer based in Adelaide, who are looking to use their skills in developing and manufacturing new tools. And the first one of those, or one of the first, is the Biopen. I have a prototype in my bag that they've done the first manufacturing run of the Biopen. And so it all starts to come together, and you can see why. I mean, so we've got this network and community of researchers and clinicians, and so it's a ready-made opportunity for manufacturers, because not only do we present them with the ideas and with the science and the protocols, but with the customer base as well, the customer base that's engaged, that wants to use these technologies, and in fact has been involved in developing those same technologies. So I want to just go back to the importance of that clinical applications. We've talked about materials, we've talked about machinery, but none of this would have been possible without being able to work with a wide range of clinical collaborators. And let me again pay tribute to Professor Graham Clark, because it was Graham who many years ago, before many of our other colleagues recognised the opportunity in this area, it was Graham who recognised the opportunity and who really drove not only the science with us and the clinical applications, but drove the establishment of this network. Some of them I've already introduced to you. But let me introduce you to a few more recent ones who've come along because of the vibrancy of this network, because they can see that fundamental advances can be turned into something that they can use in a reasonable period of time. And of course, that's the real driver behind the clinical need and the clinical desire to be involved. This is an application of 3D printing that was brought to us by Professor Stuart Mackay. He's an ENT surgeon in Wollongong. Stuart treats a lot of sleep apnea patients, and his idea was, well, from medical imaging data, can we 3D print and recreate that patient's own airway so that we can study how that collapses on the bench, and he can design the most appropriate intervention on the bench. So that's, on the face of it, sounds pretty easy, because replicating the shape of an airway is not a problem. But how do you replicate the distribution of mechanical properties? Well, first of all, it's a challenge to the medical imaging area, because we need to be able to get imaging data that reflects the distribution of mechanical properties. And we're very close to doing that. And then you have to be able to 3D print so that you can distribute the mechanical properties along the model of that individual's airway. And we've been able to do that, and we've been able to set up experimental protocols on the bench where Stuart can look at 3D printed airways and start to understand mechanisms of their collapse. Now that immediately, of course, impacts his surgical practice today, but it also gives us a platform to test very innovative things on the bench. We don't have to go to the patient to test different ways of keeping that airway open. We can develop new technologies and print it with this airway on the, and test it with this airway on the bench. Another recent example comes from Dr. Pale Mukherjee from the Royal Prince Alfred Hospital. And again, Pale approached us about the need to 3D print ears. And again, the challenge on the face of it seems okay, because 3D printing the shape or the replica shape of an ear is not a challenge. But of course, what we need to do, and the reason she wants this is because she treats a lot of kids with microtia that have a defect, which means that some of the ear or all of the ear is missing. Some of the other machinery is still intact, but you'd like to be able to put a living ear back onto that patient. So the first thing here is that, again, it's a bit like this sleep apnea project. You know, the shape of an ear, fine, but you know, the ear is fairly complex. You want to run your fingers, use your own ear. If you want to run your fingers around it, you'll see that it's quite complex. You know, there's a real distribution of mechanical properties throughout there, stiffness, softness, and then of course that's all. And you're trying to replicate that in the scaffold. So what we've been doing is building, first of all, understanding how we can do that. And then we're building a customized printer, which enables us to give the structure, print the gel materials so that we can generate cartilage, a bit like the knee project, but generate cartilage in that living ear. And it's been complex. It's been a lot of very fundamental science and engineering and understanding and modeling the distribution of mechanical properties in the ear and how we can get those mechanical properties and yet still maintain the ability to grow cartilage throughout that ear. And you can see that a lot of these applications, like the cartilage, this is going to require a dedicated bioprinter eventually to do it. So commercial printers that we see today are printers that do a lot of things with a limited number of materials, but we're starting to see the emergence of new opportunities in building customized printers for particular clinical applications. Another example of that is with a surgeon who's located right next door here, Professor Gerard Sutton, the Sydney Eye Bank. And again, Gerard approached us about developing, he's seen the biopen for cartilage regeneration. He had some ideas around a pen that would facilitate corneal regeneration or heal ulcers in the eye that might result from damage or from surgery. We've been developing with him and his team, developing a bio ink which has all of the right properties to do that. Of course, each ink has its own particular requirements, and this one has the added requirement that it needs to be transparent. So it has to have all of the mechanical properties, all of the biological properties, but now it also needs to be a transparent material that we can deliver through the 3D printing approach. So we've done that. Gerard and his team have developed most of the ink. We've assisted in that formulation development as well. And it's very much our charge is to develop the iFix pen, which you can imagine needs to be much higher resolution. There's a few different requirements in this pen compared to the cartilage pen. First of all, much higher resolution, and the way that the inks and the components of the inks are delivered are actually quite different as well. So it's necessary to have all those skills together. You know, the clinical need, the ability to formulate the ink and understand how the cells will behave in that ink, and then to develop this customized bioprinting solution for that particular challenge. And the final application, or the final one I'll mention, you'll notice there was a long list on there, is with Professor Toby Coates at the Royal Adelaide Hospital. Toby's involved in islet cell transplantations to treat diabetes. These are cells that are obtained from a donor. So they're very precious cells, and you don't want to lose any. Now, unfortunately, the protocols used at the moment do result in loss of those very precious cells during the transplantation process and immediately thereafter. So we've been working with Toby to develop a 3D printing protocol that will enable those donor islet cells to be encapsulated in a 3D printed structure, surrounded with appropriate biomolecules or perhaps other cells as well that will support their survival and fight off will counter any immune response after the implantation. So it's a very complex project, multiple cells, multiple materials, multiple bioactive components. But we've made some progress. We've developed an ink that can print the islet cells and some of those supporting cells, as well as the biomolecules. And we've delivered the very first prototype of that multi-head printer to the Royal Adelaide Hospital in just November, December last year. Of course, it's an iterative process. This is just the first prototype, and I imagine it will take us a year, maybe 2 years before we have that final printing protocol and 3D printer dedicated for islet cell transplantations. But all of the skills and infrastructure are in place to deliver something like that. I'm going to finish with something that's a bit more challenging. That's all incredibly exciting, and working with these clinicians has been a real privilege and an honour. But you know, clinicians also ask quite probing, fundamental questions. I've mentioned already Mark Cook and his interest in epilepsy, and I've mentioned Professor Xufeng Huang and his interest in schizophrenia. And of course, we're trying to use our electromaterials to treat those diseases. Their fundamental question was, could we use our recent advances in materials and fabrication to understand more about how diseases like that develop? So can you print neuronal networks on the bench, study their development, and where it goes wrong, see why it goes wrong, and how we might intervene in that? So we have done some work. We've developed ink to print human neural stem cells in recent years in 3D structures. We've also developed an ink which prints induced pluripotent stem cells. Now, this is more relevant to this study because these stem cells would be your stem cells. These are cells that are generated from cells from the skin, for example, reprogrammed to induce pluripotent stem cells, then 3D printed, and we look at the development of neural networks from those stem cells. So we can do that with a healthy individual, and we've done that. We've shown that we can generate different types of neurons through that or after that 3D printing process. But of course, we could also do it, and this is our current challenge, with the cells from diseased individuals. So induced pluripotent stem cells from patients that have epilepsy or schizophrenia. And how does that neural network develop in comparison to how a healthy neural network develops? And we're still a long way from making real significant advances in this area, but you wouldn't even dream of taking this challenge on 5 years ago. You know, the fact that we think we can get fundamental knowledge out of practical advances in processing and materials and fabrication is really quite an exciting twist in this whole story. And so we continue to develop these protocols to get different types of neurons from 3D printed structures. And again, it's such a fundamental advance. You know, there's so many studies still in research laboratories around the world that look at cells in 2 dimensions. I mean, the world is not flat. You know, I mean, biology is not flat either. And so you wonder about the relevance of those. And so every month you start to see publications that show that behavior in 3D structures is very different from behavior in 2D structures. And we just published in a parallel study, just use one example recently. This is a neural network in 2D, cells grown on a bench and cells grown in 3D. What we're doing is looking at the firing patterns, the electrical firing patterns coming from those arrangements of cells. And you can see in 2D that it's pretty noisy, stochastic response. But in 3D, we start to take on this patterning that you would see in real situations. So there's a lot of things happening in parallel that suggest to us that maybe the brain on the bench is not such a wild idea, that we can start to get some understandings. And we can use it, of course, for other things, not just for the disease modeling and understanding it, but for testing interventions, for testing new electrodes for electroceuticals to treat epilepsy and schizophrenia, for traditional drug testing on the bench, for tissue regeneration, regenerating some bits of the brain that might be implanted, and even maybe for testing. Let's go right back to our graphene electrodes. We know they interface with nerves. Can we use that to interface with nerves to drive a neural prosthetic? Well, we can start to test that on the bench with the 3D neural networks. So at the moment, we use 3D printing to print a prosthetic, a hand that has all the flexibility of a human hand, all the movement of a human hand. And it's driven by wearable electrodes on the skin at the moment. And even that's pretty good. But we need new electrode materials to keep that interface stable with time and enhance its performance. And eventually you would like to drive it both ways, to drive it neurally and to get information back into the neural system. And the ability to 3D print is enabling all of those studies. Let me finish with a thought that says that our real advances in this area won't be limited by the science and technology or the clinical drive to need it. But there are other non-technical issues which people within our Centre of Excellence are looking at. That is the appropriateness of healthcare systems, regulatory issues. How do you regulate something that's personalised? And so you're making a one-off for each individual. And of course, tied up with all of that is the social acceptance in this amazingly rapidly progressing area of technology. And we can get it all to work, it all works, but one cog stops and we're all buggered. So let's hope that we can get it all to work. Let me thank all the guys at Wollongong who do an amazing job, an amazing group of people I have the privilege to work with at Wollongong and indeed around this country and the other nodes of our Centre of Excellence, and indeed collaborators all around the world who share our vision that we can take and merge advances in materials, cell biology and fabrication, couple it to a clinical need and deliver real clinical outputs as quickly as possible. And that's what drives us all to do it. Gets us out of bed in Wollongong at 4:30 every morning to come up here and talk to you and then go back and do some work. But it's great. It's an amazing time to be doing research. This is what we're doing. These are the guys that are going to make a difference, the guys that are 25, 30 years old in our laboratories at the moment, who have all of these tools, all of those networks at their disposal. I really look forward to what they're going to achieve in the next few years, because the ability to create new systems and new structures is finally back in the hands who need to use it, the creative people who can develop new materials and new systems that will benefit us all. If you're interested in 3D printing, we have a free MOOC, which starts on March the 5th, massive open online course. Have a look at that. It involves all of our clinicians as well, which is great. They talk about their experience in the use of 3D printing. Once again, thank you very much for getting up early, coming out to have a listen to what we're all about, and really appreciate your attention. Thank you.
Thanks, Gordon. That was really very, very exciting talk. Really exciting. We've got some time for some questions. If anyone would like to ask a question. Yes?
Actually, Gordon, Rob Burford from University of New South Wales. You touched upon the question I was going to ask about the Frankenstein effect and the social acceptance issue. I'm on your side with all of this brilliant technology, but there will be strong active lobbyists out there opposing any of these sorts of changes. I'm just wondering, do you have staff who assist you in trying to manage these things?
Yeah, we do. We have a group within the Centre of Excellence which is charged with looking at ethics, policy and public engagement issues around all of these developments. And we need to continue to resource that and to work tirelessly to make sure that we bring all of the community with us. And it's a challenge because, as you've heard, the developments are quite rapid. So working in the field, it's sometimes hard to keep up with what's happening, but that's a challenge we must undertake. I think in days gone by, scientists and engineers might have thought it was somebody else's challenge. I think this generation of researchers accepts that it's their challenge, that they need to be engaged in it, and it's the only way that all the good things about this technology will be realised in those communities that we're working for.
Hi Gordon, I'm Kathy Connell from Johnson & Johnson Innovation. Thank you. It is indeed an exciting time. I'm curious about one of your second or third last slides, and you showed your breadth of engagement around the world. And in fact, I was thinking before about this being 3D printing being the ultimate connector and platform of integration and connection. But I did notice a conspicuous absence, that you don't have any collaborators in the US. And I wondered about that.
Maybe one of the arrows went askew. We do
so you
actually
have them into Canada, but you didn't have them into the US.
Yeah, and we do in Canada as well. And look, we do have collaborators in the US, but I would say that not so strong as the other collaborators around the world. And there's reasons for that I'll be happy to talk to you about later. But we do have some very good collaborators, particularly in the nerve regeneration area with University of Texas at Dallas and with University of California. So there are nice collaborations, but in terms of big organisational collaborations, there are some challenges.
You mentioned the use of stem cells quite often. Does the usefulness of stem cells deplete with a person's age? For example, if you get the stem cells from a very elderly person to use it on that person, does the usefulness of those stem cells diminish? Thank you.
I'm not a stem cell biologist, but I have been told that that is the case. Sorry.
Oh yeah, thank you. So when you put things in a human body, it's often hard to know exactly what's going on. So with all this advanced technology, are you looking at or are you incorporating the ability to do real-time monitoring diagnostics?
Yeah, definitely. Two challenges there. One is the development of contactless, non-destructive characterization tools for the things that we're making, even before implantation. So there's some very active programs in that area. Yeah, and then developing techniques and continuing to refine imaging techniques that allow us to monitor and characterize the progress of performance after implantation. There are two areas that I didn't talk about today, but they're very active around the world.
Round 7. Your map isn't quite to scale.
Really?
Curiously, you're closer to the centre of influence. I'm not quite sure which one, Sydney or Canberra. Anyway, my question was, I've commercialised a few things in my life, and I saw 10, 20 opportunities. As our metal manufacturing diminishes almost to zero, how do you see that way going forward? And it's a message for all the people involved in future industries in this country. Yeah,
look, I must admit, struggled until recently to get the traditional manufacturing sector in Australia to even have a look at what we're doing, because there's no doubt that the skills are there that can be transferred into this particular area, particularly in the, not only in the hardware end of things in terms of the printers, but even on the material processing and the ink formulation. Those skills are around. They've been used in other areas. And it's only been recently that people have started to think, oh, maybe we really can transfer those skills. So I'm hopeful. Again, we're conscious that we need to be driving that opportunity, driving the conversation. And I hope that things like the biopen and the manufacturing of it sort of highlight, you know, that here's an automotive component manufacturer who's joined forces with the research and clinical network to generate something that they wouldn't have thought of generating just a few years ago. So I think we need more and more examples like that.
Thank you. Question regarding the schizophrenic testing, the Petri dish view. Were the researchers using electron flow or magnetic field stimulation?
No, it was direct electrical stimulation, so pulsed stimulation, charge balanced pulse stimulation, but very similar to what's used in the cochlear implant.
President of the Royal Society of New South Wales. Geez, you've come a long way from smart ladies' underwear, I think.
I thought you were going to comment on the beard.
We will explain that later. Could I ask, how small can you go? So we start talking about nanotechnology, but we jump to 3D printing of micron size, millimetre size things. What's the smallest thing you think you can actually get down to?
In terms of feature size with printing?
Yeah.
So with the types of printers that I've showed you today, it is tens of microns, maybe down to low micron. There are other techniques of 3D printing that can get lower than that, but then of course, so you compromise resolution with scale. So that's where we are at the moment, and I think so. There's still room for improvement here. There's you know, what's a scalable printing technology that can get down to really high resolution. So not all of the challenges have been solved. There's still plenty for all those young guys to do, and look forward to them doing it.
I'd like you all to join me in thanking Jordan for a fantastic presentation. Thank you. Thanks very much. It's been fantastic. So the next presentation is on the I'm going to put my glasses on, 9th of May, and it's Professor Justin Gooding, who'll be talking about how sensors are changing our world. Justin Gooding's from the University of New South Wales. So thanks everyone. Those leaflets are on your seats also. So thank you again, and see you on the 9th of May.